This tutorial shows how a Spark investigator can segment, annotate, and curate their image data, and then map these data to an organ scaffold to visualize and compare them with other datasets mapped to the same scaffold. The Spark investigators are generating biological research data, which is made publicly available on the Spark portal. Data undergoes various stages, like creating and submitting the dataset in Pensive, the Spark data management platform, getting the data curated, releasing the data under embargo, and eventually making the data publicly available. In this pipeline, researchers have access to the MBF bioscience software tools to perform fair image segmentation to prepare their data for scaffold registration. For example, investigators put microscopy image data into context by adding metadata about their sample, like which organ and location the sample is derived from, the age, and the sex of the subject, which will help others understand their data. It's essential to add fiducial annotations with markers that will allow scaffold mappers to more accurately fit your data into the common coordinate framework of the 3D organ scaffold. After accomplishing each stage of this image segmentation to scaffold registration pipeline, the data is now ready to be explored on the Spark portal. Now, we will briefly explain the segmentation process for microscopy image data from the Spark dataset titled Multicolor Adeno-Associated Virus Sparse Labeling and 3D Digital Tracing of Enteric Plexus in Mouse Proximal Colon. To start, we collect the confocal 3D image data visualizing enteric plexus in the mouse proximal colon, then segment and annotate microscopy image data using NeuroLucida 360 Spark mode. First, we add fair file-level subject metadata, proximal colon tissue from female mouse at 9 weeks, then reconstruct and trace neurons, nerves of interest in MBF Biosciences open and specified neuromorphological file format, cells of enteric plexus intraganglionic nerve endings. Lastly, use the Spark vocabularies provided through API connection to SciCrunch infrastructure to segment and annotate anatomical features and identify fiducial points or regions for scaffold registration. This 3D segmentation data is provided for scaffold fitting and registration through Pensive. We have established a workflow to handle the mapping of data obtained from the flat preparation of organ samples. Here, we will demonstrate this workflow using the image data derived from the same dataset that we used in the previous example. We will walk through the pipeline with the image data derived from subject M11 in the dataset. So following segmentation and annotation of the image data with MBF's NeuroLucida 360 tool, we will use the information of where the sample was obtained from to map the segmentation data to its location on the flat coordinates of the colon scaffold. As the data is now embedded in the colon scaffold, we can transform the data from the flat coordinates field to the 3D coordinates field. This will bring the data from the flat preparation to its correct location on the 3D colon scaffold. For this Spark dataset, the investigator has segmented and annotated digital tracings for 7 subjects. We have applied the mapping workflow to all 7 tracings and mapped them to the proximal section of the mouse colon scaffold. As there are several regions on the scaffold map, uncheck the Display All option and gradually turn on each component to better understand each of the annotated regions. A map data sample can be explored in 2 ways. First, to isolate a particular sample, you can choose to pan, rotate, and zoom to bring it into view. or you can uncheck Display All and turn on the specific sample you want to look at. The Fit to Window button on the right corner can then be used to bring the sample data into full view. You can then select the colon again from the legend to bring the outline of the colon scaffold back. The thumbnail on the top right corner is used to get a gross idea of where the sample is located on the scaffold, as the orientation of the scaffold in the thumbnail is now synchronized to the scaffold in the main view. The circular muscle layer is turned on to demonstrate that we have mapped the data to the interface between the circular and longitudinal muscle layer on the scaffold. Note that exploring these features to their full potential requires access to the new functionality coming to the portal over the upcoming months. This dataset depicts individual neural wiring patterns in Enteric microcircuits and aims to showcase how neuronal soma and fibers in a myenteric ganglion interact. To achieve this aim, the investigator has created individual groups of interesting neurons. To see these specific groups, uncheck M11 from the legend and select, for example, neuron 123 connex. Furthermore, due to current portal limitations, the regions are all presented in a flat list, and it can be quite challenging for a user to identify which group belongs to which sample. Hence, we are working on introducing a tree hierarchy structure for the different regions. This will allow the samples to be presented as the parent and the groups as children of the parent sample in a tree structure. With this feature, we also anticipate better control, enabling us to toggle between the view of different groups. We are also currently working on introducing a contextual card to scaffold maps, and we believe having a contextual card with a description of the scaffold map will also allow us to capture information on the relationship between the sample and its respective groups. Lastly, our portal team is working on improving graphics rendering on the portal, and we hope to be able to present these tracings with lines instead of points in the near future. We aim to improve the portal visualization as each portal features becomes available. Mapping data to a scaffold allows any image data, be it a neuron network or vascular image, to be mapped onto a common coordinate framework for visualization, exploration, and integration in a 3D environment. Data from different experiments can now be brought together onto a common platform, and investigators now have the opportunity to explore how different datasets can interact with one another, and this helps foster potential collaboration to further our current research of the autonomic nervous system. This highlights the beauty of scaffold registration, as we now have access to a platform for investigators to continually improve and update models of nerve organ interactions as discrete pieces of data continue to come together on a common framework. Here is an illustration of the heart dataset, which follows a similar curation process of image segmentation to 3D scaffold registration before it's published on the Spark portal. While the colon and heart dataset have served as examples for demonstrating how data is mapped to the organ scaffold, this process is not limited to these two organs. Similar techniques are also being used to map data onto our stomach, lung, and bladder scaffolds. Visit the Spark portal at sparc.science, a rich public resource showcasing all this cutting-edge information and tools for advancing bioelectronic medicine.